Welcome to our deep dive into the intricate dynamics of cryptococcosis, specifically how it affects individuals with compromised immunity. Today, we're exploring groundbreaking research into how the interplay between immunosuppressive therapies and antifungal treatments influences the disease's trajectory. Cryptococcosis, driven by the pathogen Cryptococcus neoformans, presents formidable challenges, particularly in those with weakened immune defenses. It demands sophisticated therapeutic approaches to manage effectively. The study we're discussing investigates the dual pressures of corticosteroid-induced immunosuppression and antifungal therapy, using a sophisticated murine model to unravel the complexity of host-pathogen interactions. These insights are crucial, as understanding the effects of immunosuppressants like dexamethasone on fungal growth, alongside the impact of antifungal drugs like fluconazole, adds a critical layer to our treatment arsenal. Why does this paper matter? It delves into the heart of pathogen adaptation, focusing on how these drugs shape the immune landscape and fungal behavior. This research underscores the importance of fine-tuning therapeutic strategies to balance immune suppression and pathogen eradication, ultimately enhancing clinical outcomes. Let's examine the experimental design. The researchers employed a well-validated mouse model administering dexamethasone and fluconazole, which allowed them to observe changes in immune responses, fungal adaptations, and lung integrity. This methodological approach provides foundational insights into how treatments influence biological processes at a cellular level. Key findings reveal that while corticosteroids promote fungal growth by dampening immune defenses, fluconazole helps preserve lung architecture against damage. This dance between treatment benefits and risks highlights the need for nuanced strategies in combating cryptococcosis. Other findings, such as alterations in fungal capsule size, chitin remodeling, and macrophage polarization, provide a detailed picture of pathogen adaptation in response to these pressures. Strengths of this study include its comprehensive mechanistic insights into the intersection of host, pathogen, and drug interactions. The robust use of an animal model, coupled with detailed immune and pathogen analyses, allows for a nuanced understanding of these complex dynamics. However, it's important to consider the limitations. Translating these findings directly to human contexts poses challenges, and variability in individual immune responses still requires exploration. Additionally, the focus remains primarily on pulmonary effects, with broader implications yet to be fully evaluated. Looking to the future, it's clear that extending this research into clinical settings is imperative. Understanding long-term impacts and exploring various antifungal combinations will help customize treatments for better efficacy. Moreover, broader studies are required to fully appreciate the scope of these medicinal interactions. In conclusion, integrating knowledge of fungal adaptation dynamics under combined pressures provides a promising pathway for refining therapeutic strategies. This work encourages collaborative efforts across medical and scientific disciplines, forging a more precise path to effective treatments for cryptococcosis. Through careful adjustment and innovative research, we can potentially offer a glimmer of hope for patients battling this complex infection. Thank you for joining our discussion today.